right, well, first of all, thank you to the organizers for the wonderful conference and the chance to come to this beautiful part of the world, which I have never explored before, so I'm very excited to be here. And also, thank you to all of the speakers before, both for widening my mind and thinking about things I don't normally think about, but more importantly, in this particular morning session, for doing all of the introduction to the microbiome so that I can move pretty quickly through the first part. But I will say that since Emily, I don't know if you noticed, but behind her person, she had a tree. And so since she was using trees to introduce her talk, I'm going to use humans to introduce my talk on plants. So I really love this schematic because it encapsulates Emily's idea that she introduced about the importance of the microbiome in terms of just sheer numbers, and the fact that, you know, the snack you just ate, you should be asking yourself, who did I feed, me or my microbes? And you might not notice in this picture, I really should have zoomed in, but the reason I particularly love this illustration is because it includes bacteriophage viruses. And a huge part of my research is thinking about how these viruses of bacteria shape the microbiome in terms of its diversity and stability. And I'm very passionate about that and not going to talk about it at all today. So apologies for that, but I had to draw the line somewhere, and I decided to be faithful to the title of my talk, and I'm sorry, that means I excluded my human virome work, but I'm very, very happy to talk to anyone who's interested in phages in the microbiome, as that's actually the more active component of our research program. So the microbiome, whether it's humans or plants or anything else, or the ocean, it's an incredibly complex system. And that means that it requires all of our brain power, all of our different approaches, all of our different expertises to understand it. This is not something that anyone can do alone, let alone anyone, even researchers from one country can do alone. And so this is the sort of call, especially to the trainees in the audience, that if this is of interest to you, this field is a super wonderful playground to play in. So you should join us. In my lab, we take multiple approaches to understanding the microbiome, like many others, and this is very also nicely introduced by Emily this morning. One of the ways that we think about it is to take a top-down approach, which is to let the data tell us what is interesting, and just to collect as much data as you can and see what it's screaming at you. And here's some data that's not our own, but one of my favorite ways to think about this is to think about diet and fiber. From the last talk, thinking about diet impacts on the microbiome. And you can do this in a few different ways, right? You can do this by simply using a 16S amplicon sequencing and saying, what impact does fruit and vegetable intake have on the microbiome? And it's not particularly stunningly striking in this figure, but believe me that in this paper, they did find a statistical effect of fiber uptake. And you know, from the last talk, from this talk, if you take nothing else away from today, it's eat more fiber. All right, so that's one way you can do it. And then if now we're in this age where you can go far below, deeper than 16S sequencing. And so a nice follow-up study here looked across different species and actually looked specifically at fiber-degrading genes, and so these enzyme gene families, and could show that when you actually start separating the microbiome in terms of gene family, rather than just who's there, then you start seeing very striking effects of diet. So this is just a nice illustration of this sort of top-down approach, where you embrace the complexity and ask your key question. The other way you can, of course, do this is by a bottom-up approach. And so you could do this in terms of breaking up the parts and trying to put them back together and rebuild your function of interest or your pattern of interest. And a nice example of that is from Casey Huang's lab, a recent paper where he has been building a synthetic human gut microbiome. And one of the things he does is takes individual human gut isolates, grows them in a sort of gut-like media, and then afterwards spins out the bacteria, we call that spent media, and then grows a second species in that same media. And says, does the second species grow worse, better, the same, depending on who was there before it? And one of the neat things that's recently come out of his lab is that based on these pairwise interactions of how each species impacts one another, you can predict how they'll grow in triplicate. So you do a really nice job of thinking about how these might affect future or more complex interactions. Now, I have no idea what happens when you go above triplicate. I can only imagine it turns into a total mess, but we'll see. The third thing that we do in my lab is apply theory and develop theory. And I think this is, in the microbiome field, a particularly important way to study the microbiome because there is so much data, and really anything you do impacts the microbiome is my takeaway from being in this field for a little while now. But that doesn't necessarily mean it's important. And so asking the questions in a hypothesis-driven way is really critical, but hard to do. And actually, in the microbiome space, there's very little theory. And so a lot of what we do is build new theory, but also read the disease ecological theory, plant community ecology theory. Think about the existing theoretical predictions that you can use to ask questions specifically, to have a priori predictions. And so I'm going to start today by just giving you two little examples of the theory that we've been building in a system-agnostic way, which is why I've started with humans here, because our theory should be equally applicable to humans, mice, plants. And then when I get to the empirical work, I'm going to introduce the system we work on, which indeed is the plant phylosphere above ground. This is Jess Metcalf. She's one of my closest collaborators, and we get together very often and figure out what we don't know and what we might be able to better understand or at least build predictions for using mathematical models. And so as a nice example of this, this is an older model that we built to answer a question about how the microbiome might shape host evolution. So it's no surprise, we heard just a moment ago a really beautiful talk about the fact that the entire human evolutionary history, and indeed the entire history of all eukaryotes on Earth, has occurred in a microbial soup. Okay? And so there is no such thing as host evolution in the absence of a microbiome. It has been there since the beginning, and of course, it makes perfect sense that host evolutionary processes have been inextricably shaped by the microbiome that's there. But then the hard part is we don't have any comparators. We don't know what a host evolved in the absence of the microbiome might look like. And this is where theory becomes very important. And so the first theoretical question that we wanted to ask is, we know that the immune system plays two critical roles. It keeps out pathogens and learns about non-self and allows hosts to rapidly defend itself against invading microbial species, but also balances the good microbes, if you will, the commensal microbiome that's critical to host health. And so one really critical question is, like, where does the immune system draw this line? Because the reality is there's a continuum between the pathogens or the potential pathogens and the commensals, especially when you start thinking about what they might look like to the host. And so where you draw this line, how vigilant you decide to be, is going to shape both how many microbes you let in, the diversity of your microbiome, and how well defended you are against pathogens. And this trade-off shapes the fact that we are not super vigilant, but also not too tolerant. And this is particularly interesting right now because this, we think, and this model attempted to show, that where you draw this line is going to be shaped by your evolutionary history and both the microbes and pathogens that you've evolutionarily interacted with, which means that in this moment of craziness in the world, of everything changing, of the global change that's occurring on the planet, everything that we've adapted to might be rapidly changing. And so a lot of the disease we're observing is the fact that this line, if you will, has been drawn by a historical evolutionary interaction that is now very different. Not just for humans, but also for all the plants that we grow in agricultural settings and many other species. So I won't spend too much time on this, but if you're interested, please read the paper. But the key thing really was that this is shaped by many different factors, including how costly the immune system is, how many pathogens are in the environment. But what I want to call out here specifically is where you draw that threshold is also dependent upon whether your microbiome is itself part of defense against pathogens. So if having a microbiome allows a host to defend itself against pathogens effectively through competition, microbe-microbe competition, then the host can afford to be a whole lot less vigilant than it would be otherwise. And that's what we show mathematically. I have not put any equations on these slides. I apologize if you've come for equations, but I promise we have plenty in the papers. Okay, so that's one of the punchlines. And so this basically tells us that even our immune system, which we think of as a fundamental component of evolutionary optimization, is likely very critically shaped by the microbiome. The second question is, why on earth would we, as an organism, depend on our microbes when we could do something ourselves in terms of genetically encode it? And so for this, we developed this coined this term. It's not particularly effective, but MyDOT, we call it, the microbiome-dependent ontogenetic timing. Because the more we read, the more we realized that most organisms on Earth make critical life history decisions: when to flower if you're a plant, when to pupate, how quickly to grow. They make these decisions based on their microbial associations. And in some ways, this seems really a stupid thing to do, right? Because you could absolutely make a mistake and become maladapted, not make a critical life history transition because the microbe you don't happen to associate with it. And yet all plants and animals seem to do this, or at least where people have looked. And this led to the next question, which was why and when would you depend upon microbes? And so the punchline of this paper, and again, if you're interested in this, I very much suggest you pick up the paper and read it. But we basically find, theoretically, through the mathematical models and also through reading the literature and the examples, that you should depend on your microbes if they can give you information that your genes could never give you, information that you could not pick up as an individual organism. And for example, local host density as a plant, how much you grow, how quickly you grow, depends a lot on the local biomass, which, I don't know if you've noticed, but plants don't have eyes. And so microbes might act actually as a critical signal for local host density that allow plants to make this critical decision. So that's one example. So I chose these two vignettes specifically because they highlight the way that a microbiome might shape host evolutionary trajectories. If you can depend on your microbes, that means that you don't have to have that costly trait yourself, but it can be a bit of a trap. If you depend upon your microbes, then you really need these microbes. And indeed, sometimes we think having the microbiome might slow host evolution, paradoxically. Because if you're defended from a pathogen by your microbiome, that actually reduces selection acting on the host genome itself for defense genes. All right, so that's the sort of theoretical body of work that, yes, it absolutely makes sense that host evolution can be critically shaped by the microbiome, and is. And then this leads us to the empirical question: can we test these ideas? What are we looking for? So can we find evidence that host evolution was shaped by its microbiome? And to do that, I work on the plant microbiome. However, most people, when they say that, are talking about the rhizosphere below ground. And this is a very important plant microbiome. Has lots of studies associated with it. And I think of the root microbiome or the rhizosphere microbiome very similar to the human gut microbiome. It's critical to digestion, to uptake of nutrients. But the microbiome we work on is actually the phylosphere above ground. And this is a particularly understudied microbiome, and its equivalent, I would say, is the human skin microbiome. It's the aerial interface of the plant, of the organism. So there's a key role of the microbiome in protecting against pathogen invasion. And we've known about that role of the phylosphere microbiome for some time, but the more we look, the more we realize it's fundamentally important in many other ways as well. I'm just going to highlight that 60% of the terrestrial biomass on Earth is the plant phylosphere. And so this is not just an important plant microbiome, but a really critical microbial habitat on Earth. None of those reasons is why I chose it as a study system many years ago when I was setting up my lab. Really, I am an ecologist, a microbial ecologist, and I like to know where things start and stop. And it's almost that simple. When I was reading a lot of the marine literature, like you heard about from Flora's beautiful talk, and my mind just cannot comprehend how would I ever decide where a population begins and ends. And same below ground in soil. And so I looked at leaves and I said, at least I can say That is one leaf, and that is another leaf. And nobody can argue with me. And so more or less, that's why I chose this as a study system many years ago. It's spatially explicit, so it allows us to understand community assembly processes because we know what the boundaries are. But also, it turns out these are quite easy to manipulate as microbiomes. You can spray microbes onto leaves. You can wash them off very easily. There's clear trait variation across space and time. And to us, very critically, the leaf microbiome is made up of highly culturable bacteria. The leaf is basically a petri dish. So if you get your conditions right in your petri dish, you can culture nearly all of the microorganisms on there. And there's an important function. And I will not spend much time on the important function beyond the two that I'm going to highlight today, but know that the more we look, the phylosphere microbiome is critical to ecosystem processes across the globe. So what kind of questions could we ask empirically to determine whether this theoretical effect of the microbiome on host evolution might be true? And I've just chosen 4 vignettes to show you today, and I'm going to go quite quickly through the data. So apologies for that. I'm going for breadth rather than depth, but please come and talk to me if you would like to know any more about these studies. So the first question we might ask is, do we find host-associated microbiomes actually confer a fitness benefit to their host? Are these microbes critical to host health? Seems a pretty straightforward question and has now been asked across many systems. We might then ask, are hosts adapted to the microbiome? If they're so critical, what are hosts doing to not just be an open petri dish? Right? How are hosts specifically selecting for microbes? And then similarly, how and can microbiomes adapt to their host? And finally, do hosts depend on their microbiome for critical traits? So in other words, are the traits, going back to Emily's talk, are they inherent in the genomes of the host organisms, or are they outsourced, if you will, to the microbiome? All right, so I'm going to give you examples of the bottom-up and top-down approaches we've taken to answer some of these questions. And we have mainly focused on the tomato phylosphere because it's agriculturally important and also quite a nice model system. And this is Eli Melferber, who was a graduate student in the time and is now a postdoc at Georgia Tech. And Eli spent the first almost 3 years of his PhD building a synthetic community. It's hard. And he recently published a primer on how to build a synthetic microbiome in Nature Microbiology. If you're interested, you can search his name in Google Scholar and you'll find it. It's really excellent if you're thinking about embarking on this. Just note, it's hard. And there's no right way to do it. You have to think carefully about the types of questions you want to ask and build your microbiome accordingly. But Eli went for both simplicity and phylogenetic breadth. So through huge amount of culturing, he came up with 16 members of bacteria, taxa, that broadly represent the diversity that we observe in tomato growing in the field in the phylosphere. And then he created this 16-member cocktail. And generally, what we do in these experiments is we create the cocktail, inoculate plants when they're young, and we spray for the first few weeks, and then we stop spraying. So after that, we allow the plant to just naturally go, and the microbiome to naturally do what it's going to do, and then measure our traits of interest. So the first one I'm going to show you is we did this in a greenhouse setting where we inoculated tomato with this phylosphere microbiome or not, in unsterilized soil in an open greenhouse. Well, not open to the world, but in the sense of the plants were mixed, whether they were control or applied with the microbiome. And 2 things to say before I show you the critical results. The first of which is that when we did 16S sequencing, what we find is that in the control plants that did not receive the phylostart, is what we call it, our synthetic community, there's a lot of bacteria there if you look at relative abundance with 16S amplicon sequencing. And the light shades are bacteria that do not belong, they aren't from our phylostart community. So they were things that were just flying around the greenhouse or maybe in the seed or in the soil. And at first we were kind of worried because that looks like a pretty diverse microbiome to some extent. And you compare that to our phylostart applied plants, and we were happy to see that our synthetic community does make up the majority of the microbes we find on the leaves. But nonetheless, does this actually matter? Maybe this is enough. So then we went to droplet digital PCR to do absolute quantification of bacterial abundance on the leaves. And when you use droplet digital PCR, what we find is that, yeah, there's some diversity there in the control plants, but in terms of actual biomass, in terms of actual cells on the leaves, these plants are essentially gnotobiotic. And you compare that when we actively apply the phylosphere microbiome, only then do we see appreciable amount of bacteria. And this was critical because it allowed us to ask the question we wanted to ask, which is, does this microbiome matter? And the answer was yes. And the first trait that we measured was actually how many tomato the plant produced at the end of its life. And I won't spend the details here. These are different fertilization treatments. But in each case, we saw that the PhyloStart applied plants had greater yield than non-treated plants. And this was exciting. It was the first time a link has been made between the phylosphere and plant fitness, in other words, the amount of seed produced. And so we were quite happy about that. But also, and building on many other studies from many other systems, these phylosphere microbiomes were also disease protective. So if you look at Pseudomonas syringae, a common bacterial pathogen, we find less of the bacteria of this pathogen in the presence of the microbiome. So at least two critical traits here, disease protection and growth, are associated to this above-ground plant microbiome. So yes, host-associated microbiomes confer fitness. So that suggests then that plants should kind of care if they get them or not, right? So to that next question, we took a different approach, this sort of top-down approach, where we went out to the beautiful botanical garden here at UC Berkeley, and this work was led by Venka Smets and Kama Chak. And what they did is they chose 20-odd plant species and swabbed the leaves in a common garden environment and asked a really simple question. Do they have the same microbiome? They're all growing in the same place. So if it's just about dispersal, they should have the same microbiome. And the answer was no, don't spend too much time on this plot. The plot itself is not particularly useful, but the statistics tell us that about 21% of the variation in the microbiome is explained by which plant species it is. So certainly, even in this common garden environment, the plants are selecting. How? We're still not sure above ground. However, we had one additional clue, because Venka and Kama did something kind of fun, which is they separately sampled the top of the leaf and the bottom of the leaf. And the bottom of the leaf is where all the stomata, the pores are, where the plant breathes and also releases leaf exudates. And what they found was that the top surface of the leaf had much more diversity. And I'm not showing you the neutral model here, but we did some neutral community assembly modeling and could show that the top of the leaf is basically just whatever lands there. The bottom of the leaf is where you see these endemic species. So these were the plant species-specific bacteria. They tended to be found on the bottom of the leaf. So this gives us a clue that yes, plants do filter or select for their microbiome, and they seem to be doing it through exchange of potentially exudates or other volatile compounds and so forth. So far, so good. Is the microbiome adapting to the plant? And the other really nice top-down approach you can take is you can just take a microbiome and select upon it and see if it responds to selection. And I'm showing you just a teeny portion of the huge data set that Kyle Meyer, who was a postdoc in the lab, generated. But he took this experimental evolution approach by starting with a microbiome from the field, from tomato, and taking this one pool of microbial taxa and selecting them on either tomato, a wild Pimpinellifolium tomato species, pepper, or bean. And you just grow them on that plant, wash them off at the end of the experiment, apply them to the next plant. You can do this in replicate. And you can ask through experimental evolution time or through multiple selection rounds, do you end up with a different microbiome if you've been evolved on bean, then on pepper, then on tomato, even though you started as the same original microbial inoculum? And again, I'm just going to show you one tiny punchline here to convince you that yes, this happens. At the end of the experiment, what Kyle did is he took a microbiome that was evolved on, for example, bean, and he put it together with a tomato-evolved microbiome, crashed the two communities together. We call this community coalescence. sprayed it onto tomato, and asked who won. Okay? And so another way of saying that is, if it's evolved on tomato, is it going to outcompete something that evolved on bean, for example? And so what he found is if you do these experiments, so if you just inoculate a tomato plant, in this case, with a bean-evolved microbiome, it looks very, very different than a tomato plant that receives a pepper-adapted microbiome. So the first punchline here is in this very short experiment, 6 passages in the greenhouse, we saw rapid shift of the microbiome depending on which plant host species it was evolved on. But the really neat thing is then let's crash together tomato and bean. All right, that's this sort of other kind of pinky purple here. When you do that, this is just a pure tomato microbiome. The tomato microbiome wins. It outcompetes the bean-adapted microbiome, and it outcompetes the pepper-adapted microbiome. So in these head-to-head competitions, these well-adapted microbiomes absolutely win out. They outcompete the species that were adapted to pepper or bean. So to the third question, and I'm running out of time, but I think I can make it. To the third question, absolutely. In such a short period of time, these microbiomes can adapt, and this is probably mainly through species sorting, different taxa dominating, but potentially this was over 6 months in the greenhouse, so we're currently doing metagenomics. It could also be through mutational change and adaptation. All right, let's get to the last, I'm gonna come back to this at the end, actually. All right, so to the very last question, does the host then depend on the microbes? And so for this, we actually switched to Arabidopsis, simply because the genome is slightly better annotated. And what Eli did is he applied the same synthetic microbiome, the tomato synthetic microbiome, to Arabidopsis, and then challenged Arabidopsis with a common pathogen, Pseudomonas syringae again. And for today, I'm going to have you mostly ignore the polyploids, except to just say that in polyploids, the microbiome didn't matter much. But in diploid Arabidopsis, what we found, just like I showed you previously in tomato, is that when the syncom is there, you get less disease. Fine. But in this particular case, Eli did a bunch of transcriptional analysis, RNA-seq. And what's really neat is that when you look at the genes that are differentially expressed in the presence of the microbiome compared to control, the microbiome was priming the plants, if you will. So the plants upregulated more defense genes in the presence of the microbiome than plants that did not have an intact microbiome. And so we were able to show mechanistically that, that indeed the plant is relying on the microbiome in at least 2 ways. One, I didn't show the data, but the microbiome directly outcompetes the pathogen. Right? So there's competition on the leaf surface. That's a direct impact. And then indirectly mediating all of these beautiful defense genes that, of course, themselves evolved in this microbial soup. So these two, the immune system and the microbiome, are, again, intricately linked. Here's, if you're a plant person, I know there's at least one in the audience who might be interested in the exact genes. Here they are. I think this gene in particular looked familiar from Dominique's talk earlier in the conference. But anyhow, we see specific genes that are upregulated only in the presence of the microbiome. All right, so I'm out of time, but that's perfect, because one thing I really want to make sure I finish with here is that none of these stories are complete. I gave you some just tiny snippets to convince you that this is a worthy field of study, and also that the plant phyllosphere is a fun playground to ask these questions in. But I want to end with these huge open questions. And so the first one is, does the microbiome matter primarily under stress or only under stress? And does this make sense in terms of my dot theory? And so what I mean by that is simply, I haven't shown much of the data here, but we tend to see that the microbiome is particularly important in disease protection. It also confers more growth to the plant, but only in really bad conditions. So if the conditions are great, then the host actually doesn't need its microbiome nearly as much. It's in this more stressful conditions that the microbiome becomes important. And that is what the theory predicts, the MyDot theory predicts, that you would outsource things that might be important only under certain environmental conditions, not all the time. And that seems to be what's happening. And a word of warning that, again, because we're in this moment of unprecedented global change, this is where we're going to start to see the microbiome becoming particularly important in increasing host adaptability, and yet it's also at a time when microbial diversity on the planet is decreasing significantly, or seemingly so, and so we might be breaking all of these host microbiome interactions exactly when they're needed most. Will any microbiome do? Or is there an advantage of having a specific, well-adapted one? I showed you in Kyle's experiment that the microbiome rapidly adapted to be bean-like or pepper-like. But what I didn't tell you is there was no fitness effect on the tomato. So the tomato itself didn't care if it got a bean-adapted microbiome or a tomato-adapted microbiome in terms of the fitness effects we could measure. So this is really an open question and an important one. As microbiomes adapt, how much of that is due to adaptation to the host versus microbe-microbe interactions? So we did some network analysis in that experiment, and we could show that the microbiomes, as they're adapting to pepper and bean, the whole network structure changes remarkably. And that suggests that it might be adaptation to bean, but it could also be that something about bean is changing the microbial interactions, and that's actually what's important. And then finally, I showed you we have this probiotic now, or this synthetic consortia that could be used to increase tomato yield. But the question of how and when we should use that is a really important one. We heard a beautiful talk about invasive species during this conference. And as we move forward, as we're developing these probiotics, I think we need to be careful and really be thinking about whether we're creating these microbial invasive species and invasive taxa and what impact that might have on resident microbiomes. So with that, I will thank all of the folks in my lab who've helped with this and my collaborators who I thanked along the lines, and take questions. Thank you very much.